Hello everyone, my name is Jessica and this is my presentation on genome-wide characterization of genome in a bottle HG002 benchmarks dataset. So here's a little background on my presentation for today. Whole genome sequencing, also known as WGS, allows us to identify genetic variation across the entire genome and has become an important tool in genomics research as well as precision medicine. Because sequencing and variant calling methods can produce different results, reliable benchmark datasets are needed to evaluate the accuracy of these analyses. One really common and widely used benchmark is the Genome in a Bottle HG002 dataset. And this dataset contains millions of validated genetic variants and serves as a reference, also known as a truth set, for evaluating sequencing analysis. And although this dataset HG002 is well established as a benchmark resource, my project focuses specifically on characterizing its genomic features used for exploratory data analysis. This led my research question, which looks at what characteristics of HG002 support its use as a benchmark resource for whole genome sequencing analysis. And just to continue on my research question and objective, after introducing the importance of benchmark datasets, I wanted to determine whether the GIAB HG002 benchmark dataset demonstrates the genomic characteristic expected of high-quality benchmark resource. So specifically, I asked whether the dataset provides a broad genomic coverage and sufficient representation of all major variant types used in WGS analysis. To answer this question, I used exploratory data analysis to evaluate chromosome level variant distribution, variant composition, chromosome-specific variant density, and genome-wide variant positions. So I hypothesized that HG002 would contain validated variants across the genome and substantial representation of both SNPs and indels, supporting its use as a benchmark resource. For this study, I used an exploratory data analysis approach to characterize the GIAB HG002 benchmark dataset. I first began by obtaining the HG002 variant data and preparing it for analysis. The dataset was then organized in a way that I could examine the variants based on chromosome, variant type, and genomic position. Then I performed several descriptive analyses. First, I made sure to calculate the total number of variants identified on each chromosome. This allowed me to be able to examine how variant counts were distributed across the genome. I then classified the variants by type, specifically separating SNPs from insertions, deletions, or indels. I also calculated the proportion of each variant type within the dataset. And because chromosomes differ in size, I also examined variant density rather than looking at raw numbers of variants. This helped me be able to consider the number of variants relative to chromosome length. Lastly, I examined the genomic positions of the variants to visualize their distribution across individual chromosomes, and I determined whether variants were broadly represented throughout the genome. The analysis was primarily descriptive and summarized using counts, percentages, variant density calculations, and some graphical visualizations. Together, these methods help me be able to characterize the dataset from several perspectives: how many variants were present, what types of variants were represented, and how those variants were distributed across the genome. And then the next few slides, we'll be able to walk through the main findings of these analyses. So just to reiterate, I did use the GIAB HG002 benchmark dataset, which provides a highly characterized set of small genomic variants. One of the strengths of this dataset is that it includes variants across all autosome and sex chromosomes, giving us a genome-wide view rather than just focusing on a specific region or gene. The dataset also contained both SNPs and indels, which are two major types of small variants examined in this study. This is important because a useful benchmark dataset should represent different types of genomic variants across the genome. And overall, HG002 provides a very well-established reference dataset that allowed me to be able to explore how validated variants are represented and distributed across the genome. So here we have figure 1 from my data, and figure 1 shows the distribution of validated variants across each chromosome in the GIAB HG002 benchmark dataset. Variants were identified on every autosome as well as chromosome X and Y, demonstrating a broad genome-wide representation. Although chromosome 1 contained highest number of variants and chromosome Y contained the fewest, you can see here in the charts, every chromosome was represented, which supports one of the key characteristics expected of a benchmark dataset for WGS analysis. So here we have figure 2, which is our variant composition of SNP and indels. In figure 2, we are looking at the composition of variants in HG002 dataset, and specifically the proportions of SNPs compared with indels. And as we can see here, SNPs are made up at 79.82% of variants, while indels only accounted for roughly 20%. So it's clear that SNPs were clearly the dominant type of small variant in this dataset, and it represents almost 4 out of every 5 variants. The difference is important because SNPs and indels represent different forms of genomic variation. SNPs involved in a change in a single nucleotide, while indels are involved in the insertion or deletion of one or more nucleotides. And although SNPs are much more common, the dataset still contains substantial representation of both major SNP variant classes. This is relevant for a benchmark dataset because variant calling pipelines need to be able to identify different types of genomic variation, not just the most common type. So the predominance of SNP is also consistent with what we generally expect in a human genome, where single nucleotide variants occur more frequently than small insertions or deletions. So the main takeaway from this figure is not simply that there are more SNPs than indels, but that it shows HG002 contains millions of variants from both major small variant categories, with SNPs representing the majority of the dataset. And it supports overall the characterization of HG002 as a dataset with a substantial small variant diversity that can be used when evaluating WGS analysis. So here we have figure 4. In figure 4, we are looking at SNP and indel distribution across chromosomes. And figure 4 takes the previous analysis one step further by looking at how SNPs and indels are distributed across individual chromosomes. Here we see that both SNP and indels were identified on every chromosome, showing that two major small variant classes were represented throughout the genome. And SNPs also remain the predominant variant type across chromosomes, which is consistent again with the overall variant composition that we saw previously in figure 2. The important finding here isn't necessarily that the difference in the number of variants between chromosomes, but it's that both variant types are consistently represented across the entire genome. So this helps provide additional support that HG002 contains the broad genomic representation and variant diversity expected as a benchmark resource for WGS analysis. Overall, the dataset showed broad representation of validated variants across the genome, including all autosome and sex chromosomes. It also included substantial representation of both SNPs and indels. However, SNPs were the predominant variant type. One important takeaway was that variant counts and densities differed across chromosomes. These differences are expected, however, because chromosomes do vary in size and biological characteristics such as gene density and recombination patterns are seen differently in all chromosomes. So therefore, these differences should not necessarily be viewed as a limitation of the dataset. When considered altogether, these findings demonstrate that HG002 actually contains the genome-wide representation of small variant diversity expected of all genome benchmark resource. So rather than evaluating how well HG002 performs as a benchmark, this study helps characterize what the dataset actually contains and how its variants are distributed. It also helps provide useful context for understanding why HG002 is an established resource for whole genome sequencing and variant calling studies. There are several limitations that are important to consider when interpreting these results. The first being the study focused on single benchmark genome, HG002, because I did not compare HG002 with any other GIAB genomes or other benchmark resources. These findings are specific to this dataset. A comparison with additional benchmark genomes could help determine whether the patterns observed here are consistent across different individuals or datasets. Second, my analysis was primarily exploratory and descriptive. I focused on characteristics such as variant composition, chromosome level distribution, variant density, and genome-wide patterns. And because of this, the study describes what is present in the dataset rather than testing why those patterns occur. Another limitation is that I only focus specifically on small variants, including SNPs and indels. Structural variants and other more complex forms of genomic variation were outside the scope of this project. Including those variants in future analysis could provide a more complete picture of the HG002 genome. And it's also important to recognize the differences in variant counts and density between chromosomes can be influenced by biological factors such as chromosome size, gene density, and recombination patterns. So these differences should not automatically be interpreted as strengths or weaknesses of the benchmark dataset. Lastly, I did not directly evaluate the performance of a sequencing or variant calling pipeline. Because of that, my findings characterized HG002 as a benchmark resource, but they do not demonstrate that HG002 is superior to other benchmark datasets or establish the performance of a particular variant calling method. So these limitations also help provide opportunities for future research, including comparisons with additional GIAB genomes, analysis of structural variants, and direct evaluation of variant calling performance. In conclusion, this project allowed me to characterize the GIAB HG002 dataset and to better understand the genomic variation it represents within this widely used benchmark resource. And overall, I believe that the findings did answer my research question by being able to show that HG002 demonstrates the genomic representation and small variant diversity expected of a whole genome sequencing benchmark. But most importantly, this project highlights why it's important to be able to understand the characteristics of the data being used for benchmark. Before a dataset can be used to evaluate sequencing or variant calling methods, we should be able to understand what types of variation it contains and how that variation is represented across the genome. While this study was descriptive, it provides a foundation that could be expanded through comparisons with other benchmark genomes or through direct evaluation of variant calling performance. The main takeaway from this project is that HG002 does provide a very well characterized and diverse representation of small genomic variation, supporting its established role as an important resource in WGS sequencing. And that concludes my presentation. Thank you guys for watching.